embryonic cells? Well, the human cells that are really helpful for using in the lab.
So there are a lot of times when scientists are going to want to do experiments in human cells. These could be either to test a drug, to see what a gene does, to make a protein that can't easily be made in bacterial cells, as we'll talk about later. So there are these various reasons why you want to do experiments in human cells. But if you were to just take most human cells, just like a primary tissue sample, and try to get it to grow in cell culture, it wouldn't be very happy. Maybe it would divide a couple of times, but then stop. Because cell lines are typically mortal. Have a finite lifetime. They're not just going to grow indefinitely. However, there are also cell lines that are immortalized. And one of the main ones that we use are these HEK 293 cells. So, what exactly are these cells? So these cells were originally isolated in 1973 by a Dutch scientist named Alexander van der Ebb, and then they've been further adapted. So he isolated these HEK cells from kidney tissue of an embryo. The exact origin of this is not quite clear, whether it was aborted or spontaneously miscarried, and I'm not going to get into any of that, so don't go there. But anyway, this is one of the main cell lines we used, and typically use these HEK 293 cells. So basically, these were generated in the 293rd experiment by a postdoc in this scientist lab. So a postdoc, that's someone like me. So it's somebody who had recently gotten their PhD and now is doing research, additional research, often typically in a different lab to try to get new skills, etc. And so the scientist, this postdoc Frank Graham, what he was doing was he was constructing these cells with adenoviral DNA. So adenovirus, this is a double-stranded DNA virus. And basically, he was taking this sheared adenoviral DNA and he was transfecting or transforming these cells with it. And so when we talk about transformation, that's basically getting genetic information into cells, and there are different types of ways you can get to do this. But he was transforming these cells with this adenoviral DNA with the sheared DNA. So basically, not the whole adenovirus, but broken up pieces. And these pieces, some of the pieces would integrate into the host genome. So basically, they would insert themselves into the host genome. And if they did this just the right way, then they could actually get these cells to potentially grow forever. So he wasn't trying, this is what happened, was that this gene fragment, about 4.5 kilobases, so about 4,500 DNA letters long, piece of that virus inserted itself into the chromosome 19 of these HEK cells. And it did so in such a way that it disrupted um, and it introduced these genes from the adenovirus, these E1 genes, E1A and E1B. And basically, what these did was it shuts down the quality control system that the cells have over over their cell cycle control and apoptosis. So basically, your cells have a lot of mechanisms in place to prevent them from becoming immortalized because that would cause things like cancer if these cells grow out of control. And in fact, that's why this scientist was originally doing these experiments, was to try to study how certain viruses can cause cancer. And so this stretch of DNA that had this viral proteins E1 and A and E1B, basically these proteins are able to mess up the machinery that the cell has to try to keep control over its cell cycle. So over its production of DNA, its replication, its copying of all of its DNA, and then its division, splitting that DNA into the daughter cells, etc. And basically, cells sometimes have this mechanism called apoptosis, which is the programmed cell death, where things get out of control. The cells kill themselves to prevent things like cancer from happening. And these viral genes were interfering with this process and allowing these cells to replicate indefinitely.
E1A also activates various viral promoters. So if you have a protein under a promoter like a CMV promoter, then the E1A will help you get large levels of that protein expressed.
And so this original integration was important, was essential for allowing these cells to grow indefinitely. And so the 293rd experiment, it had this perfect integration where you get those genes and you got this and stably integrated. So these cells were able to stably make the viral proteins over time and maintain that and grow indefinitely. So this is where the original HEK 293 cells came from. And then they've been further adapted. So one of them you might hear about is HEK 293T. So the T does not stand for transfection, although these cells are really good for transfecting. So basically for getting genetic information into them. So I talked about how we can use that term transformation for getting genetic information into cells. We typically use that term more commonly when we're talking about like bacterial cells. And when we're talking about human cells and other types of cells, we typically use this term transfection. So they were transfecting these cells, we can transfect them, we can stick genetic information into them. And this can be really helpful if we want to do things like express the proteins. We can stick in like a plasmid, it's a circular piece of DNA, and then we can stick the instructions for, say, making a protein. And then we could get the cells to make that protein. This is a common method we use. This is recombinant production. So we've recombined the gene with this plasmid DNA, and then we stick it into cells. We transfect those cells with it. And so this is something that we commonly want to do when we are studying what various proteins do. We want to put a reporter in there. There's various reasons why we want to transfect cells. And the HEK 293 cells, or the T cells, are really good for this. But one reason that makes them really good is that they're able to stably maintain the plasmid. So if you stick the plasmid into the cells need a way to actually make the plasmid. So when the cells copy their DNA, they need to be able to copy the DNA of the plasmid as well. The thing that specifies like replication are like origins of replication. So these ORIs. These HEK 293 T cells, they express a temperature-sensitive version of the T antigen from the virus SV40. This T antigen is this protein of this virus that allows it to control its replication. And if you get the cells to make this SV40 T antigen, then those cells can cause that plasmid that you stuck in there to be replicated. If you put that plasmid, you make the plasmid have that origin of replication. And so this will allow you to keep that plasmid growing in those cells. But if cells don't have that T antigen, then a virus that contains, if a virus doesn't have a T antigen protein, and if the cells that make it if the cells you put it in don't have it, then the cells can't replicate that plasmid, but they can take in that plasmid. And so this can be used in gene therapy as well as like viral vector vaccines, where basically you're able to make a lot of virus that is unable to replicate itself in anything other than these special cells that have those proteins that are needed. And so say you make a virus, you want to then be able to transfect cells to get these cells to like transiently express it. Maybe this is, so just express it for a while, things like that. Maybe you want to use, so maybe it's like a vector vaccine. So you want to get cells to make this protein, and you can't just like genetically engineer these cells because these cells are going to be in people's bodies and things. And so basically, if you have the virus, then you take out that T antigen, and then you stick it in these HEK 293 T cells, which have that T antigen, then the virus is able to replicate. You can make the virus and then use it in other purposes, and do similar things with like the E1. And because those, like with the HEK cells, they have those E1 proteins. And so things like this are allow you to make viruses that can be used in the lab safely because they can't replicate outside of cells that don't have these the missing viral components. And but you still, because these cells have the viral components, they have to be treated with special precautions. So you take like BSL-2 precautions of biosafety level 2. When you're working with these cells, you're working in a hood, you're using sterile conditions. Although a lot of the sterile conditions are so that you protect the cells, because the cells, you don't want them to get contaminated by like bacteria or fungi and things like that. And so you also have to, it's important to periodically check the cells, make sure that they're actually the cells that you think they are, make sure they're not contaminated by like mycoplasma and various things. So those are like the HEK293 T cells, the ones that have that T antigen. But then there's these Treg cells. So basically, these have a tetracycline repressor gene. So we've talked about bacterial inducible expression before. Basically, you want to make a protein on demand. Bacteria typically do this. You can induce expression using like IPTG, and so you have like a repressor, and then you add IPTG, the repressor falls off, you're able to make your protein, blah blah blah. So the basic idea, though, is that this TET repressor is going to allow you to do something similar with the addition of the antibiotic tetracycline in these cells that have a TET promoter. And so basically these cells are going to make this tetracycline repressor gene, and therefore you're able to use this tetracycline repressor inducible expression system in these Treg cells. Okay, so these cells these are about being used. Often we're talking about them being used as adherent cells. So basically you're growing them in dishes, or you're growing them in these flasks. And in all of these cases, these plates, and in all these cases, the cells are actually stuck to the bottom of the plate. And then when you want to, they're eventually going to overgrow themselves, and then you split them up. You add like trypsin to kind of break up the proteins, break up the cell-to-cell contacts, and break up the cell-to-plastic contacts. And the first what you do is you like suck off the liquid. So the cells are stuck to the bottom of the plate. You suck off that liquid, call it aspiration. Suck off that old stuff, and then we add fresh media, a little bit of fresh media, and then we trypsinize them if we want to take them off of the plate. So we don't want them to like overgrow to start. We need them to have plenty of space to grow. So typically when they're about like 80% confluent or whatever, so basically they cover about 80% of the surface area of the plastic. Then you add trypsin and you chop them up. Chop up the cells, but just like the outer cellular connections on the cells, and then like put up a little bit of media. And then count them, see how many live cells there are, and then calculate how many you need to like seed on a new plate, so that you'll have they'll have plenty of room to grow. And so this is what typically you use when you're doing adherent cell culture. There are HEK cells that have been adapted to be used for suspension cell culture. So this is when we're growing them in like flasks, similarly to what we would do with the bacterial cell culture. So HEK293S, these are suspension cells, and then there are different versions of these suspension cells. So there have been, often when we're dealing with these HEK cells, we're growing them in like this media that is not chemically defined. So it often is this base media, but then you add like fetal bovine serum FBS, which can vary from batch to batch and all of these various things. And so there's also defined media. So basically these like serum-free media and things like that. And these freestyle 293 and these XB293, these are like Gibco, I think. They're expression systems. These are really good for expressing recombinant proteins, just like we were talking about bacteria. But here we're talking about more large-scale protein production. So we're talking about like suspension cells inside of flasks. And these two cell lines are going to use different like defined media, so like the freestyle media or the XB media. More about that on the sites of those groups. But these cells are supposed to be good for that. And then these are just kind of like basic 293 cells, but they've been adapted to grow in the specific media in these suspension cells. There's also like specialized versions of these So one of the reasons why people want to produce proteins in human cells as opposed to like bacterial cells is that human proteins, if you're trying to make a human protein, especially if it's a complex one, the protein might have a lot of like post-translational modifications. So things like glycosylation and phosphorylation. So the instructions for making a protein are written in DNA, and then you get messenger RNA copies, and then the bacteria and like insect cells and stuff, they'd be able to make a protein, but they might not be able to make a protein properly. And so you might get a protein that's not going to fold correctly because there are proteins called chaperones inside of cells that help proteins like fold correctly. And then also proteins might need modifications. They might need post-translational modifications, things like phosphorylation or glycosylation, so the addition of sugar chains. And bacteria and yeast and insect cells, they might not have this machinery. And so if you want to make a protein that has modifications, insect cells can be good because they're more similar to human cells than bacteria, but they have slightly different versions of proteins of the modifiers. So you might get different modifications. So then you typically want to change to a mammalian expression system. And so typically this would be like CHO cells, so Chinese hamster ovary, or HEK, P93 cells. So those are the two common cell lines that are used for mammalian protein expression. So even if you're in one of these lines, though, there can be a lot of variation in the glycosylations that a protein gets. So there are various different sugars that can be added, and they can be added in these different ways, in these different forms, and they can also be added in different places. And so N-linked is basically saying that it's getting attached to asparagine residues. It's just what the N stands for. But you can have different like branches and all this different stuff. And if you're doing something like structural biology, where you're trying to get a picture of the shape of a protein, if that protein has a bunch of different glycosylations, that can prevent you from getting a clear picture of the protein because it's all going to be different. So you want something that's homogeneous. And so there's the cell line, this HEK 293SG, and it's good for generating these homogeneous proteins, homogeneously glycosylated proteins, because it has mutation in this MGAG1 gene. And basically, it's going to prevent it from adding a certain modification. So when it can't add that modification, it can't make those really complex sugars. And so it makes these simpler sugar modifications, and this makes it good for various purposes, like doing the structural biology of like a membrane protein or a secreted protein. And it's also like a TET-inducible protein version. So yeah, some final notes. Let me just make sure I haven't. Okay, some final notes is that in terms of this paper is really interesting. They're looking at like the genome dynamics of these cells and like how these cells, like the genome and how it responds to various things. So these cells are going to be highly unnormal. And so when we talked about how these cells can grow like indefinitely, well, as they're growing indefinitely, remember they've lost that like quality control thing, which is why they're able to grow indefinitely. That also means that they can keep acquiring new mutations. And these mutations can then prevent them from acting like normal cells if you're trying to study normal cells. And they can also cause like experiments that you do early on and like in an early passage. So when you haven't split them that many times versus like a late passage when you've split them a lot of times, you could have like huge differences in the results. And so that's not good. And so you typically want to use as close to the early passage as possible. So when you get that early passage, you then like store stocks of them, and then you don't passage them too many times. Instead, you get a high passage number, then you go back and you take a new stock vial and you thaw it out and you split it and you keep growing it. But anyway, so even with that, these cells are highly abnormal. And they're what we call like triploid. So basically they have triploid. So basically they have about 3 copies of each chromosome, but not quite. So they have the modal number. So the one that's most common is about 64 chromosomes. But some of them have more, some have less, etc. in terms of growing them. So you typically grow them at like 37 degrees Celsius and 5% CO2. The ones with the shaking, the suspension flasks, you need to keep them constantly moving. So these can be shaker platform, or they're like bioreactor thing like spinner flasks where they have little spinners in them, as well as like roller bottles and things like that. But often the suspension cells are grown at 8% CO2 instead of 5% CO2. The color of the media is going to indicate whether like kind of like the health of the cells and what the CO2 levels and things like that. So you don't want that media to get all yellowy. And yeah, that's basically, they double about every 34 to 36 hours. And I'll post links to some helpful resources and articles. Hope that helped you understand these cells, and yeah, happy experimenting.